Hello, my name is Joshua Mugabe. I welcome you to cell division. What is mitosis? What is the importance of mitosis? Mitosis is the process in which one eukaryotic cell divides into two cells identical to the parent cell. Generally identical, since alteration of genetic material can occur, more or less organelles may be distributed between the daughter cells. Mitosis is fundamental for asexual reproduction of eukaryotes, for the embryonic development, for the growth of pluricellular being, and for tissue renewal. Why, in some cases, is mitosis synonymous of reproduction? In some living being, asexual reproduction occurs by many means: binary division, schizogony, budding, grafting, and many others. In asexual reproduction of eukaryotes, mitosis is the mechanism by which the constituent cells of the new beings are made. The term mitosis does not apply to prokaryotes, since it involves nuclear division and eukaryotic structures. What is the importance of mitosis for embryonic development? Every embryo grows from a single cell that suffers mitosis and generates other cells that also divide themselves by mitosis, forming tissues and complete organelles. The perfect regulation and control of each of these cell divisions are fundamental for the creation of the normal individual. Without mitosis, the embryonic development would be impossible. What are some examples of organs and tissues where mitosis is more frequent, less frequent, or practically absent? Generally, in vertebrates, mitosis is more frequent. In tissues that require intense renewing due to their functions, like epithelial tissues and the bone marrow. In plants, the meristem tissues has numerous cells undergoing mitosis. Mitosis takes place with low frequency in tissues of slow renovation, like the bones in adults and the connective tissues. In some adult tissues, mitosis is almost absent, like the nervous tissue and the striated muscle tissues, or what we call skeletal and cardiac. The nervous tissue develops from stimulus by development of new electrical networks between cells and the striated muscles. Tissue grow by cellular hypertrophy. How does mitosis participate in the growth of pluricellular organisms? All procellular beings grow with increase in the quantity of their cells. This increase is produced by mitosis. Although some types of growth occur by cellular hypertrophy or by deposition of substances in the interstitial spaces. What is the uncontrolled mitotic process that occurs as disease in procellular beings called? Uncontrolled mitotic cell division is called neoplasia. Neoplasia, the formation of new strange tissues, occur when a cell suffers mutation in its genetic material, loses the ability to control its own division, and failure of or is transmitted to its descendants. Cancer, a malignant neoplasia. The term malignant means neoplastic cells can disseminate to distant sites, invading organs and tissues. Neoplasma whose cells cannot disseminate to distant sites are called benign neoplasm. Is the internal epithelium of the bowel the same as it was 1 month ago? The internal epithelial covering of the intestines act as a protective barrier and also as a means of nutrient absorption. The traffic of ingested material inside the intestinal lumen is very intense, and consequent tissue damage requires incessant epithelial renovation through cell division. The tissue renovation is completed in 2 or 3 days and is made by mitosis. What is cellular regeneration? How is mitosis related to this process? Some tissues are able to regenerate when injured. The liver is an example. Regenerates when small pieces of hepatic tissues are removed. Bones make new tissues in fractured regions, etc. Some animals, like Planalia, are capable of regenerating their bodies when sectioned. Tissue regeneration, cellular proliferation happens by mitosis. What is cell cycle? Cell cycle or mitotic cycle is the time period that begins when the cell is created and finishes when it is divided by mitosis, creating two daughter cells. The cell cycle is divided into interphase and the mitotic phase. In cell division, happening during the entire cell cycle, what is interphase? Cell division properly occurs during mitotic phase of the cell cycle. During interphase process, that are preparation of cell division take place, like the duplication of DNA and centrioles. Interphase is the preceding phase of the mitotic is the following phase. What are the three periods into which interphase is divided? Interphase is the preceding phase to the mitotic division. It is divided into three periods: G1, S, and G2. The letter G comes from gap, meaning interval or breach, and the letter S comes from synthesis, indicating the period in which DNA replicates. In fact, gap is not totally appropriate for the periods immediately before and after DNA synthesis. The idea of growth would be more adequate since these periods, G1 and G2, the cell is growing to divide later in mitosis. In general, which phase of the cell cycle has longer duration? The interphase comprises approximately 4 out of 5 of the cell cycle, and the mitotic phase has quite a shorter length. What are the events that mark the beginning and the end of the first interphase period? What happens within the cell in this period? The first interphase phase is the G1. It begins with the end of the preceding cell division. That is, with the formation of the new cell, and it ends with the beginning of DNA replication. In the G1 period, the cell is growing. What are the events that mark the beginning and the end of the second interphase period? What happens in the cell in this period? The second interphase period is the S. It starts with the beginning of DNA replication and finishes with the end of that process. The main event in this period is the synthesis of new polynucleotide chains, each bound to each DNA chain, that serves as a template, that is, the duplication of the original set of DNA molecules. What are the events that mark the beginning and the end of the third interphase period? What happens in the cell in this period? The third interphase period is the G2. It begins with the end of DNA replication and ends with the beginning of the first period of mitotic phase. On G2, the cell is growing too, and the duplication of centrioles occur only in cells that have these structures. Does mitosis properly occur before or after interphase? Is it mere point of view issue? Mitosis must be considered as a succeeding phase after interphase, since this is the preparation step of mitosis. Thus, it is not merely a point of view issue. Into which periods is mitosis divided? Mitosis is divided into 4 periods: prophase, metaphase, anaphase, and telophase. What are centrioles? In which type of cells are they present? Centrioles are tiny cylindrical structures made of 9 microtubule triplets. They appear in pairs in the cell. Centrioles participate in the making of cytoskeleton and of cilia and flagella. In cell division, they play a role of the formation of the aster fibers. Centrioles are structures present in the cell in most protists and some primitive fungi. There are no centrioles in cells of superior plants, and in general, it is considered that plant cells do not have centrioles. Although this is not entirely correct, since some plants have centriole containing cells. The region where the centrioles are located is called the centrosome of the cell. What are the main events of the first meiotic period? The first meiotic period is prophase. During prophase, the following events occur. Migration of each centriole pairs. Centrioles were duplicated in interphase to opposite cell poles. Aster formation around the centriole pairs. Formation of spindle fibers between the two centriole pairs. End of chromosome condensation. Disintegration of the nucleolus. Breaking of the karyotheca. Dispersion of the condensed chromosome in the cytoplasm. Binding of chromosomes to the spindle fibers. What is the mitotic apparatus? Mitotic apparatus is the set of aster fibers, radial structures around each centriole pair, plus the spindle fibers, fibers that extend across the cell between the two centriole pairs located in the opposite poles of the cell. The mitotic apparatus appears in prophase and has an important role in the orientation and grouping of chromosomes and other cellular elements, causing them to separate and migrate to the opposite cell poles. Substances that disallow the formation of mitotic apparatus, like colchicine, a molecule that binds to tubulin molecules and prevents the synthesis of microtubules, interrupt cell division. Colchicine is used to study the chromosomes since it paralyzes mitosis when chromosomes are condensed and so are more easily viewed under microscope. What are the main events of the second mitotic period? The second mitotic period is metaphase. In metaphase, the following events occur. Condensed chromosomes bind to the spindle fibers and get concentrated in the middle of the cell. There is the formation of the mitotic apparatus is completed. Metamorphosis ends with the breaking of the binding of the identical chromatids, and then anaphase begins. What are the main events of the third mitotic period? The third mitotic period is anaphase. In anaphase, the following events occur: duplication and breaking of the centromere with the separation of the identical chromatid, traction of the identical chromatid each to opposite cell pole, and beginning of the chromosome decondensation. During mitotic anaphase, is there separation of homologous chromosomes or separation of identical chromatids? In the anaphase of mitosis, the identical chromatids separate, and complete pair of homologous chromosomes continue to exist in each daughter cell. The first separation of homologous chromosomes occur in anaphase of cell division by meiosis. What are the main events of the final mitotic period? The final mitotic phase is telophase. In telophase, the following events occur: decondensation of the chromosomes, each set located in the opposite cell pole, karyotheca formation around each set of chromosomes, forming two nuclei, destruction of the mitotic apparatus, reappearing of the nuclei, and beginning of cytokinesis, the division of the cytoplasm to ultimately separate the new cells. What is the name of the cytoplasm division in the end of mitosis? What are the differences in the process between animals and plant cells? Cytoplasm division occurs after telophase. It is called cytokinesis. In animal cells, an invagination of the plasma membrane towards the cell center appears in the equator of the parent cell, and then the cell is strangulated in that region and divided into two daughter cells. This type of division is called centripetal cytokinesis from outside. In the plant cells, cytokinesis is not centripetal, since the division happens from the inside. Membrane sac full of pectin concentrate in the internal central region of the cell and propagate to the peripheral towards the plasma membrane. The pectin-containing sacs fuses themselves and form a central structure called phragmoplast. On the phragmoplast, cellulose deposition occurs and the true cell wall is created to separate the daughter cells. Plant cells thus present centrifugal cytokinesis. Phragmoplast has failures or pores to permit cytoplasmic communication between the daughter cells. These openings are called plasmodesma. Why is it important for chromosomes to be condensed during mitosis and decondensed during interphase? During mitosis, the main problem to be solved is the correct separation of chromosome sets between daughter cells. If chromosomes are decondensed, long tiny fibers of DNA would be dispersed in the cytoplasm. After the karyotheta breaking, and the chromosomes could not be easily organized and pulled by the spindle fibers. During interphase, the function of chromosomes, i.e. DNA molecules, is to synthesize of RNA and thus of proteins. For this task, it is necessary for the functional molecular region to be decondensed. These regions form euchromatin. During interphase, in addition, DNA replication occurs as a preparatory stage of cell division. In this process, it is fundamental for the exposition of DNA molecule to serve as a template to the new DNA chain and the production. How does the quantity of genetic material vary within the cell during the subsequential phases of the cell cycle? The first period of the first phase, interphase, of the cell cycle is the G1, followed by S and G2, and then by the mitotic phase. In G1, the ploide, the quantity of DNA molecules in the cell, can be represented by the formula 2N. N is the number of the DNA molecule in the gamete cell of a given species. In the S, DNA duplicates and the quantity of the genetic material increases from 2N to 4N. In G2, that quantity is constant, 4N. After the mitotic phase, the quantity of the genetic material decreases to 2N in each daughter cell. What is the difference between astro and anastromitosis? Astromitosis is that in which there is formation of the aster, a structure made by the centrioles. Anastromitosis is that in which there is no formation of the aster. It occurs in the cells without centrioles, like the plant cells, superior plants. Can mitosis occur in haploid cells and in triploid cells? Mitotic cell division can occur in haploid cells, diploid cells, triploid cells, and so on. Mitosis is a coping process that does not interfere with the cell ploidy. Concerning their final products, those are daughter cells and their ploidies. What are the differences between mitosis and meiosis? In mitosis, one cell, for example, with 2 N chromosomes, duplicates in its chromosome sets and divides, generating 2 other cells, each with 2 N chromosomes total. In meiosis, one diploid cell, 2N, duplicates its chromosomes too, but 4 cells with N chromosomes are generated. Concerning their biological function, what is the difference between mitosis and meiosis? The main biological function of mitosis is cellular multiplication, a fundamental process of the growth and development of multicellular organisms, tissue renewing, asexual reproduction, and many others. The biological function of meiosis in gamete formation, in gamete meiosis, or spore formation, in sporadic meiosis, the production of cell qualified for sexual reproduction with half the quantity of chromosomes compared to the original cells. There is a special type of meiosis that happens in the zygotes of some algae, protozoans, and fungi. This meiosis, called zygotic meiosis, has the function of reducing to a half the number of chromosomes of the adult individual that will be formed from the zygote. In species with zygotic meiosis, the adult individuals are haploid and they form gametes by mitosis. These gametes fuse in pair with others and generate a diploid zygote that then undergoes meiosis to restitute the normal ploidy of adult individuals. For the biological diversity, is mitosis or meiosis the more important process? Meiosis is the cell division process that allows the formation of gametes to sexual reproduction with arbitrary separation of each chromosome of the individual homologous pairs. These gametes can fecundate gametes from other individuals, promoting combination of homologous chromosomes from different individuals. In that manner, the chromosomal recombination provided by meiosis and sexual reproduction create individuals with dissimilar genetic patrimony from their fathers and thus promote biological diversity. Some fungi species and plants, for example, present spolylic meiosis, a structure where half of the chromosome of the species is generated from meiosis. This structure, by mitosis, forms gametes. Even in this case, diversity comes from meiosis. Meiosis then is the cell division process that, in conjunction with gamete mutations, is responsible for the biological diversity. Even in species having zygotic meiosis, the arbitrary separation of homologous chromosomes in meiosis creates biological diversity. What are the respective ploidy of gamete, zygote, somatic cell in a species with gametic meiosis? Adapting the pattern of X, quantity of chromosome for the gametes, zygote will have 2X chromosomes and somatic cells will have 2X2. Why is meiosis important for the maintenance of the normal quantity of chromosomes of a species with sexual reproduction? A reduction to half the maximum normal quantity of the chromosome is mandatory in some phases of life cycle of the species that reproduce asexually. If that could not happen in each generation, whenever zygote is formed by fusion of gametes, there would be duplication in the quantity of the chromosomes in the geometric progression. What is the difference between sexual spore and gametes? Do humans present sexual spores or gametes? Sexual spores are structures generated from meiosis with ploidy, number of chromosomes, reduction to a half compared to some spore mother cell. Spores germinate and give existence to gametophytes, individuals, that by mitosis form gametes. The meiosis that generates sexual spores is called sporic meiosis. It is, for example, the type of meiosis that occurs in plants. Gametes are also cells with half the number of chromosomes of the normal cells of the species, but they are specialized in vegetation, the fusion with another gamete that generates the zygote, a cell with a double number of chromosomes than the gamete cells. Gametes can appear from gametic meiosis or by mitosis in gametophytes originated from sexual spores. In humans, as well as in most animals, the meiosis is gametic. There are no spores, no alteration of generations. The male gamete is the sperm cell, and the female gamete is the egg cell. Is interphase of meiosis different from the interphase of mitosis? The interphase that precedes meiosis is similar to the interphase that precedes mitosis. In them, the main event is DNA replication or chromosome duplication. What are the two divisions of meiosis? What are the main events that occur in those divisions? Meiosis is divided into the first meiotic division, meiosis 1, second meiotic division, meiosis 2. During meiosis 1, there is separation of homologous chromosomes with the formation of two haploid cells. In meiosis 2, there is separation of identical chromatids of each of the two haploid cells created in meiosis 1, giving birth to 4 haploid cells. Meiosis 2 is a process identical to mitosis. In which meiotic division does the separation of homologous occur? What are the ploidies of the generated cells after the end of the process? The separation of homologous chromosomes occurs in the first meiotic division, meiosis 1. After the end of the cell division, 2 haploid cells are made, each having different chromosomes, with no sets of homologous. Note that in cells generated after meiosis I, each chromosome is still duplicated, since the homologous chromosomes and not the identical chromatids were separated. In which meiotic division does the separation of identical chromatids occur? After the end of this process, what are the ploidy of the new cells? The separation of identical chromatids occur in the second meiotic division, meiosis II. After this cell division, similar to mitosis and does not alter ploidy, the cells are still haploid. They have become haploid after meiosis I. How many cells are made after meiosis I and meiosis II? After meiosis I, 2 cells with already separated homologous are created. After meiosis II, 4 cells are created. What are the periods of the first meiotic divisions? Meiosis I is divided into prophase I, metaphase I, anaphase I, and telophase I. In which period of meiosis does the pairing of homologous chromosomes occur? Pairing of homologous chromosomes is a vital step for meiosis because the rightness of the homologous separation depends on the process. The event occurs in prophase I of cell division. What is crossing over? In which period of meiosis does this event occur? Crossing over is the eventual exchange of chromosome fragments between homologous chromosomes. The phenomenon occurs in prophase I when homologous chromosomes are paired. Crossing over is of great importance for evolution and biodiversity, since it provides recombination of alleles, those are different genes, linked on the same chromosome during cell division by meiosis. What are chiasms of homologous chromosomes seen in prophase I? Chiasms are intersection of 2 tracts to form X. The chiasms seen in prophase I are chromosome arms crossing over same arm of their homologous. In fact, when chiasms are seen under a microscope, chromatids are exchanged. Chromosomal segments with other chromatids of its homologous. is there interface again between meiosis 1 and meiosis 2? There is no interface, no DNA duplication between division of meiosis, only a short interval called diakinesis occur. What are the respective functions of the separation of homologous chromosomes and of the separation of identical chromatids in meiosis? The separation of homologous chromosomes in meiosis 1 has two main functions. Reduce half the total number of the chromosomes. Generating haploid daughter cells at the end of the process to make possible genetic recombination, since the separation is a rotary. That is, each pair of daughter cells can be different from other pair, relating to chromosome combination from paternal and maternal origin. And if crossing over is considered, each of the four resulting cells can be different from the others. The separation of identical chromatids in meiosis 2 has the same function in the mitosis. It separates the chromosomes already duplicated to the daughter cells. During meiosis division, does ploidy reduction occur? Does ploidy reduction occur in mitosis? In the cell division by meiosis, ploidy reduction occurs in meiosis 2. Initially, taking an example, a 2N somatic cell, ploidy increases to 4N, duplication of DNA during interphase. During meiosis 1, simple homologous chromosomes are separated. Ploidy falls to 2N, the original number. Then during meiosis 2, ploidy finally falls to N, the resulting daughter cells. Ploidy reduction does not occur in mitosis. This fact shows that although in meiosis ploidy decreased from its original number, in meiosis 2, the process is similar to mitosis. The cause of the reduction is that what happens in meiosis 1, that is, the separation of the homologous chromosomes. Thank you very much for participating in cell division. Please do like, share, and subscribe.